Hello everybody, my name is Rouzbeh and I'm a data scientist at MS Society Canada. Today I would like to talk to you about a paper by Jin, Braisley, and Jacola titled Junction Tree Autoencoder for Molecular Graph Generation. So I will start by explaining the context of this work. So this work was done in the context of drug discovery, and drug discovery finds its roots in organic chemistry. But what is organic chemistry? The definition of organic chemistry comes from the time when researchers actually started investigating biological systems, and they found out that in biological systems there's a high ratio of carbon, and to a lower degree hydrogen, nitrogen, and phosphorus. But what is the reason behind nature selecting the chemistry of carbon for creating biological systems? The main reason is the diversity of chemistry of carbon. In regards to drug discovery, why do we create or generate molecules in silico or using computers? One of the main reasons behind that is the cost associated with creating these molecules in labs. So, and also the time spent for creating these molecules. Another factor is the complexity of biological systems. So in biological systems, when we create a drug, we eventually want to introduce them into a biological system. And these biological systems are highly complex. So as an example, a protein has a highly complex and large structure. It is dynamic, so it can be stimuli responsive. It can change its structure based on the environment. And it can be highly specific to the targets it binds to. Another problem is that we don't have a full understanding of all of these biological macromolecules. So that makes sense to create molecules in using computers and modeling, test them, select a few, and then move on to the phase where you create them in labs. Now, I talked about creating these molecules in labs and using computers, but how do we transfer this information? So traditionally, chemists have used a variety of ways to transfer information about molecules. One of the most common ones is the UPAC name. And UPAC is basically International Union of Pure and Applied Chemistry, and they set conventions as to how chemists do different things. So they have a convention for how you name molecules, and this is one of the most basic ways of transferring information about a molecule. A much simpler way is referred to as a molecular formula. And in this way of representing information about a molecule, you basically just transfer information about the types of atoms and the number of atoms. So this doesn't include a lot of information. As a result, we switch to another way of representing a molecule, which is referred to as display formula. So instead of just showing the atoms, we also show the connections, what type of bonds are involved. Now, you can show these structures in different ways. So the structure that I have here is different than the one I have in the bottom. So in the bottom structure, we're showing all the atoms, and in the top one, instead of showing all the atoms, I'm showing some of them with just lines. So instead of showing I'm removing carbons and showing them just as lines. So here we actually have a carbon. Now, organic chemists are a bit lazy, and instead of showing all atoms, we sometimes remove hydrogens. So this structure is basically the same as this one. Now, there's another level of information that you can provide. These molecules are usually shown in 2D, but they're actually three-dimensional structures. So another method of showing the structure is they're three-dimensional structures. The last method that is very common, is commonly used for designing molecules in silico using computers is the SMILES lines, or a SMILES representation. And SMILES stands for Simplified Molecular Input Line Entry System. So in this method, you transfer information about a molecule using strings. This type of transfer of information is highly packed with information. So it includes all the information, almost all of the information that we represented here. Now, in autoencoder research, they have usually used this type of information for generating molecules. Now, there's a disadvantage to using SMILES. And the main disadvantage to using SMILES is that the majority of these modelings happen using NLP-based ideas. And in case of SMILES, despite the fact that two completely identical molecules, two molecules can be completely identical, their SMILES, the representation could be different. Now, what's the issue? NLP is highly reliant on strings and how they are formed. Now, when you end up having completely different strings and information for two identical molecules, what happens is that eventually you end up generating a lot of molecules that are not valid. So chemists had already thought about this, and in 1998, Dixon and coworkers came up with the idea that they can use tree structures to represent information about a molecule. And they referred to it as feature trees. And the reason behind that was that not only did they use tree structures, but they also packed the different parts of the molecule with information. Now, this work was basically ignored for a long time by researchers who were working on autoencoders and generating molecules. Until Jin, Braisley, and Jacola came up with their paper. And their proposal was that, why are we using SMILES? When chemists have already came up with this idea of using tree-based systems, why not we use tree-based systems and feed that into the autoencoder? So, and their idea was that by doing this, maybe the autoencoder can learn better. So their design and architecture is what I have here. Their code is available online, and they use a data set called Zinc. Their design is based on using two encoders and two decoders. The first encoder encodes the junction tree, or the tree structure, and the second one creates the molecular graph. Now, I will split this into two pieces.
So their goal is just to take the molecule and represent it in a lower dimensional space, or a simple, like, what is the goal of
generating molecules? So. Okay. Basically, the idea of drug discovery is this. You have a library of molecules. You want to create, and let's say those are known to work against cancer, some specific type of cancer. Yep. Now, you have a library of those molecules. You want to generate molecules that are similar to those. Technically, what usually ends up happening is that you have to get a chemist to sit down and test different molecules or come up with new molecules, and you test them in the lab. Right. This way, you're using model-based systems to generate them. And this reduces the number of molecules that you end up with. So their idea is to generate those molecules and generate them with properties that we want.
So why encode them?
So just a quick piece of information could help. Similar to GANs, autoencoders can also be used to generate new encodings. Well, you have the encodings, you can generate new symbols of whatever it is that, whatever your X is. So you have your X, you encode it, you get your encodings. You can use your representation to generate other X's that you haven't seen. And the way you do that is that you just put in some restricted or randomly generated embeddings or encodings. So that's why they're using autoencoders.
It's not just generating, I think if it's just generating, you can do it randomly. But I think here, like the end goal is generating the stable ones because of all these electrons.
Generating the ones that have the properties that we want. This is the end goal. So let's say you want to select the molecules that are the easiest to synthesize, that are best soluble in water, that have charges.
And so these are all features?
These are the things that a chemist has to think about when creating a drug. So all these things, even the size of the molecule, can affect whether or not it can interact with a protein. So all of these features are super important. Now I will start with the first part, which is the junction tree encoder.
Rouzbeh, if it's not too, I know you have a lot of slides,
I have
so I don't want to halt you unnecessarily. If that's the case, we'll just move on if that's irrelevant information. But I want to know a little bit more about the previous NLP approaches. So if you go to the slides where you talked about smiles,
yeah.
Just for myself to compare, I think the previous one, or next.
For the next one.
Oh, this one, yeah. So you have your molecular structure down there at the bottom of the page, and so not the string representation, right? And then you do NLP on that.
These are smiles representation. Yeah, yeah.
And you do NLP on that.
Yeah. So basically, what they do is that they try to detect the structure based on these strings, and this becomes super complicated when these two structures are very similar, but their strings are different. So that's what creates the complication. So let's say you have two words. You have two words that are similar, but the representation, the way we represent them, are completely different. So it makes it hard for the model to learn those.
So when you say NLP, you're talking about classical NLP, because I think that RNNs would smooth that away for you.
Based on just autoencoders, I know that they have ended up with results that are not super good, and I've shown that.
So it is probably like this is just a linear sequential graph
so
versus a 2D graph.
Yeah, so because previously in NLP people were using like hidden Markov models or other sequential classifiers, and I assume that's what you mean by, because RNN, I assume like sequential classifiers are what you meant by NLP, because no? So even RNNs, they've
So their ideas are based on NLP. They don't necessarily use all the, so I will show you examples of how they will use ideas from NLP, okay?
Yeah, let's go.
Yeah, I think it's just a straight on.
Yeah.
So they just borrow ideas. All right, so let's take a look at tree decomposition. This tree decomposition idea is basically comes from NLP. In NLP, we have something referred to as parts of speech tagging, and here the authors propose that we use a similar technique to chop a molecule into little pieces. So think of a molecule as a sentence, and we're chopping it into little pieces, and we're categorizing different parts of it. Now, they don't refer to this as parts of speech. They refer to it as vocabulary of components, and they introduce 4 types of categories. The first one is referred to as a ring, and a ring is a cyclic structure. So the structure I have here, the pentagon, is considered a ring. The second one is an atom, and the third one is a bond, and they also consider edges and links as part of their vocabulary. Now, I'm going to try and explain how the process works, so I will start with the molecule here. As you can see, again, I'm showing all the hydrogens. Organic chemists are lazy. We remove the hydrogens. We end up with this structure, these 2 structures. Now, here we have.
What's the difference between the edges and bonds?
What do you mean?
There's
Edges and this is a tree. This is basically a tree structure. This is not a molecule. Okay. So we end up with a structure like this. Now, N represents nitrogen. It's an atom. It has been color coded as blue. So that's our part of speech. Here. We have nitrogen, carbon. These are bonds, and color-coded green. The pentagon is considered a ring, and it has been color-coded as orange. This is just me trying to explain how the process works. Now, So basically, think of it as a sentence. We're chopping it into pieces and figuring out which category it belongs to.
Why do they exclude both C and H bonds?
C and
Because there's so many of them, by default, it's safe to assume a carbon has hydrogens
That's a
already.
Yeah.
bond between a C and H. That's just
Like if a C atom has like a one thing attached to it, everything else is an H by assumption.
Yeah, regular structure in chemistry, basically.
All right. So the molecule that I'm going to use for the slides as an example is the one on the.
Can you go back? So what do the edges mean?
So we will like the edges are basically based on tree structures. Is basically the node that connect the nodes. So we end up turning these into nodes. I will explain that later.
Okay.
Is that okay?
Sure.
All right, so we're going to use the structure on the top left-hand side of the slide. This is the three-dimensional structure of the molecule. Don't worry about these structures. If you want to talk about why they're important, we can talk about that after the talk. Now, let's start with this example. So this is the molecule that I will use as an example. What we do is that we start by numbering each of the atoms. So each of them is assigned an index. Now, based on these indices and the category they belong to, we categorize them. So for instance, 213 is the bond between carbon 2 and carbon 13, and it's considered as a bond. These are all rings. Now, if you connect all of these together based on this structure, you end up with a tree structure that looks like this. Now, this is a highly complex tree structure. The color-coded boxes with numbers.
How is it a tree?
I will show you how it turns into a tree later.
Okay.
Like it's
trust.
it's a tree with loops.
Right.
You're correct.
So are there,
Okay.
end leaves?
Just
Or
wait.
is it just a graph?
I guess we'll see.
Just call it a graph.
Graph for now, tree to be.
Tree to be, graph for now. Okay, so the color-coded boxes represent the number of atoms that are shared between these. I'm going to refer to them as nodes if that's okay.
Not sure, I mean.
Okay, with components of the graph. All right, so highly complex graph. We're going to try and figure out a way to simplify it. Now, the authors suggest that the edges that have more than two atoms, that are sharing more than two atoms, should be collapsed and combined together. So what we end up doing is for 6, 7, 8, we collapse them together.
What's the, sorry, I must not have caught it, but what's the 1, 2, and 3, the ones in the middle? Is that just the atom?
Which one?
How many atoms do you share?
The 1, 2, and,
The number of atoms they share. So I went from using molecular structure to using this graph.
I see.
Good?
Yep.
Okay, so we're going to collapse the ones that have more than two atoms shared. So 6, 7, 8 will collapse. We group them.
Yep.
And for 4 and 5, we do the same thing. Now, we're going to combine these as one component, relabel it, and then we end up with a structure that looks like this.
Wait, so you only do one at a time, one level? Like you only collapse trees at this point, even though you said you're going to collapse everything with a
I'm showing it this way because it's easier to, it's a step by step. I'm showing it as a step by step process, but the researchers do it in like one step.
So basically, you're just reducing the number of nodes and basically flattening it down to its most simplified state with as few nodes as possible that represents the entire structure.
Yeah.
That is correct.
So, and the more connections they have, the more likely they're going to be collapsed? Is that?
No, they just are collapsed if it's greater than two.
Like they're sharing something. The claim is that you're not losing much information
Yeah,
by doing that.
That's right.
Well, it's also because they have they create one of the decoders, right? Goes back from the collapsed structure to the full structure, right? So that's part of the model.
Oh, there's an entire decoder just for that step.
Anyway, go
Sorry,
ahead, go ahead.
we'll get
This is basically just referred to as tree decomposition. This,
Yes.
For all of the molecules in the batch, they do this.
Yeah.
I'm just giving you one example. They might be dealing with 7,000 molecules. Okay, so we end up with a graph that looks like this. Now, we still have loops. And the whole idea behind trying to get this to a position where it looks like a tree is to be able to transfer information. I mentioned this when I talked about the work by Dixon in 1998. So the idea is to use this as a way to transfer information. Now, let's focus on another part of the molecule, the graph. This part of the graph, we have 1, 0, and 3. Now, if you pay attention to the atoms that constitute those bonds, they actually share one atom. So 13. Atom 13 is shared by all of these. Now, the authors suggest that we introduce a new node. Am I allowed to use node? Okay, so we introduce a new node. By doing this, we remove one loop. So we end up with a structure that looks like this.
So
Now,
node represents. Can you just talk more about what the node is representing?
okay, so previously we talked about their vocabulary of components. That's how they refer to it. And their vocabulary of components included bonds, rings, atoms, and edges, right? I used, so let me go back. I used bonds, I used rings. I didn't use atoms, okay? So that's what they're doing. They're introducing this so that they can get rid of another loop.
Okay.
Good.
Just as a quick aside, I mean, really a lot of what you're building here is you're effectively constructing a grammar
Good job, yeah.
for
That's
the graph, right? So
correct.
it doesn't really matter too much about the details unless you really care about chemistry. But
It's basically,
yeah.
you should understand what I'm trying to do
Yeah.
because the ideas are the same.
Sorry, really stupid question. So I'm still kind of missing the logic behind why it's desirable to replace that ring with effectively this 6, this new entity.
So we,
It represents one atom because they all share one atom, right? So that the blue node, the 6, represents the one atom that they all share, which in this case is 13. So if you look up there, 1, 0, and 3 all share atom 13.
13, 13, 13.
And so you're presented with that one node 6 that represents that atom.
Okay.
There shouldn't one be connected to 6 then? I don't understand.
Yeah, that's another, yeah. That makes sense. So why isn't one connected to 6?
because you don't want cycles.
we're removing cycles. So the whole idea, so
So you
let me
can-
explain why we're doing this. Basically, we're doing this because we want to transfer information. If you want to transfer information and you transfer information in a loop, it's very hard for the process to converge. Okay, so that's what we're doing.
How do you choose which ones to cut off and which ones to leave? Because clearly you left 0, so why didn't you leave the one at 1?
Based on the structure. Some of them you can't just randomly remove. So if you just come up with rules that I want to do this and I want to do that, and eventually you end up with something that doesn't make sense, why are you doing that?
So these are based on chemistry rules?
Yes.
Okay.
So chemists are fine with 1 being over there, right? Just up in the air.
So it's
I'm
more
just
important.
What in the end?
It's
So
like
like
more
there is
important
a-
to have the 0 rather than to have the 1.
This one? This
Yeah.
this
No,
just
no, no.
this is where wait for the edge.
The node one.
The node one, the green one.
The green node
He's
one.
talking about the
It's
green.
fine.
The question is like, okay, yeah, you got it.
Okay, so after they do that, they do fragmentation. They basically tokenizing. So they tokenize the parts of the molecule, and you end up with what? If it was a sentence, you would end up with your tokens. Now, they convert them into smiles, and they create a dictionary. Now they have a dictionary. If they do this for, let's say, 7,000 molecules, they end up with a dictionary of the components.
Did you say what tokenizing is?
So
Just quickly.
So, so tokenizing is a basic operation where you divide your strings into tokens. So tokens would be the atomic representation of what you have. So a sentence is divided into tokens that could be words, for example. Words could be divided into tokens that are letters.
can I toss in a quick question? It's just in the conversion to the smiles at the bottom. It looks like we've lost basically all the the spatial information that was initially contained in the distribution of tokens.
I think it was the linear ordering.
Oh, don't worry about this. We don't care about how we're just collecting information about the components. So we just want to know what these components are. Well, this is the only.
Does each of these nodes
Right
have-
now, we're just collecting information. So think about it like this. We're just trying to figure out what words are in the sentence.
This is like a bag of words model rather than like some word-to-vec embedding of a. Okay, gotcha.
And none of these nodes has any message embedded in it. It's literally
Right
just
now,
representing a
okay.
bond or.
So after I end up creating, I'll explain how the process works. Just give me a second. Give me 1 second.
In the next 40 slides, you'll see.
I have so many slides. Okay, so this is what we eventually end up creating. So that's the vocab, and they end up providing a unique ID to each of the words. Okay? Molecules or components, but I refer to them as words.
So in the end, that representation is fully unique for any particular molecule? Like
Which representation?
when you go back 1 slide, so these nodes at the bottom here, and you have this identification as the vocab at the bottom?
So let's say this one.
Yes.
Is the exact same thing as this one, so it has been given the same ID. These have been given the same ID.
But it's like a per node ID per se. So if you were to take the molecule, would any 2 molecules have the exact same encoding at the end?
So right now, we're not really encoding. We're chopping them into pieces. We're figuring out what components are available in the library. So let's say you have a paragraph and you want to figure out all the unique words in that paragraph. That's what they're doing.
Okay.
Does that make sense?
Yes.
So why does 4 and 5 have different number? If it's both.
Four.
Not the same.
Their structure is different.
Yeah.
So is that so the structure is encoded, like represented in those?
There are 2 layers of structure. This is effectively, it's just like an arbitrary level of, the level, we've abstracted away 1 level of complexity,
And
and
then
then
we
we're
bring it
saying we're
back?
cutting it off there. We're not gonna do any more spatial
Okay.
resolution.
The structure of the subcomponent.
Yeah.
It's like a bag of words, but instead of words, you have vectors. So it's like a bag of vectors, and everything's encoded in vectors, but they put them all together at once. And
Not yet, but eventually, yes.
wait 20 slides.
All right, so we still had 1 loop in the graph. What do we do with that? So what they end up proposing is to find a minimum spanning tree. So basically, we had the weights, which were weights for the edges, so which equal to the number of atoms shared. They find the difference between that number and 100. Don't ask me why 100. They just do that. And basically what they do is that you add up these numbers for each of the edges and you find the path that gives you the lowest number. And as soon as you find that, you remove 1 edge and you end up with a junction tree. Go ahead.
Sorry about this, but what is precisely the distinction? When do I decide which loops to break by subtracting away an atom and which loops to break by doing the MST approach?
So it's specifically based on their design. Okay, so they're not technically. It's technically based on the paper I talked about from 1998 by Dixon. The ideas come from there, but their implementation is a bit different because specifically for the molecule I chose, that molecule was similar to a molecule chosen by Dixon, and I specifically chose that to compare how their implementation differs from Dixon's, and it actually differs. So this is their design. This is their decisions. This is how they're trying to make a junction tree.
And how did you say you're getting those numbers again from 0 to 100? Just
So
Subtract
the
the number of ones.
difference between the numbers on the edges and the grid. There was a question?
Well, I think just to clarify, they added the node 6 because there was a bunch of shared bonds between a single unit, or not shared atom.
There was a shared atom.
So I'm assuming that there is no shared atom between 0, 4, and 5.
0, 4, 5. There are shared atoms between these 2.
Right.
Not between these 2. Okay.
Well, there was one share. There was 1 bond there.
You can basically think of it like this. Let me go back. So we have 0.
right?
You're transforming that part. This 0 is basically this bond. I'm not showing it properly. It looks like this carbon is the node, but it's
Yeah.
actually this bond. So
That's a
this
double
bond
bond,
is
sure.
connected to this joint that is connected to both of them. Okay, so it makes sense to break the extra link and turn him into 4 connected to 5. Does that make sense?
Yeah, it's the grammar again. Yeah, it's fine.
Well, it depends on the type of the node because you can't break the bond.
No. So the rule is that you don't break any bonds in the actual structure. So whatever you do on the graph, if it destroys this structure, you're done. That structure is not valid. All right, so we ended up creating this junction tree. But if you think about the junction tree like a family tree, right now our junction tree doesn't have like the parents, the children, so on and so forth. So what they do is that they provide a label for each of them. So they make the proposal that the node that contains the atom 0 be given, be assigned as root. And the leaves are easy to detect. The only thing is the singleton. The singleton is assigned to the node that that has one atom only. Okay, so now you also labeled all your nodes. So this is what I was talking about. This is the amount of information that they are actually collecting.
Wait, what is special about atom 0?
Atom 0. Oh. The reason why they're doing this and assigning roots based on, it's just something that they decided to do. I don't necessarily know why. And the numbering is basically generated by a package called RDKit. I don't know if RDKit makes specific decisions as to which atom is assigned 0. It might make specific decisions as to which atoms is assigned 0, but the authors don't talk about it. They just say, we are going to make the assumption that the node that has the atom 0 can be assigned as root.
I see, so that numbering is not random, there is a logic
I
behind it.
think there's a logic behind it, but the authors basically use the package and RDKit generates these numbers for you.
Do you know what it stands for, RD? Like the RD part of RDKit?
The
Research and development kit.
R&D, but sure.
So 0 and 1, are they just intermediate nodes
Not
in
that.
the tree? 0 and 1, are they just the intermediate nodes in the tree? Like they
Yeah.
don't represent anything special?
Nothing.
Okay.
So we started with a smiles representation. We end up with this much information about the tree. I'm showing it in the pandas table, but this is actually, they don't use pandas at all, so the information is not represented like this, but they store this information for each of the molecules. Now, we have created a junction tree, but how do we don't have a path. How do we move and transfer information between nodes? So they do a tree traversal, depth first, meaning they assign the root as depth first, and they do the tree traversal. They end up with 2 paths like this. So one is bottom up, and the other one is top down. So this gives you the ability to transfer information from one of the leaves to the root, and the opposite way. And this column basically represents the depth.
Are you changing your color coding?
What am I doing? Did I?
So 4 was
Yeah, 4 was the
orange.
bond, right?
Oh yeah. I
So is it
apologize for that. I don't know how that happened.
Yeah, that's fine. Just confirm.
Is it still a bond though? Still a bond.
It's
same thing.
still the same thing. I apologize if I changed it.
That's good.
Okay. So just to see how it works. So 2 to 1, 1 to 6, 3 to 6, so on and so forth. So now you have 2 paths to basically scan the tree. Now, how do we collect information about each of these nodes? So we previously generated WIDs for each of the components, correct? So now what you can do is scan scan the tree structure, collect the WIDs for each of those components. So that's exactly what they do. So they select an arbitrary leaf, collect the WID or word ID from the dictionary, and then they transform it into word embedding. And they end up with something like this. Now, when they're running the model, they're actually using a batch. So the batch starts at the arbitrary leaf for all of the molecules and works its way up to the root. So this is basically the information for the leaf for all of the molecules in the batch. Now, I talked a lot about transferring information. What information are we transferring? I never talked about what information are we're transferring.
So before, could you go back to the previous slide? How does the? I just want to make sure I know how you're making the embeddings.
Which vocabulary are you gathering from it? The ID from the vocabulary?
The dictionary
Yeah, so like
before? The dictionary he said he would use eventually. I was using it.
So like the dictionary in the dictionary, what are the items in the dictionary? What are the keys? What are the keys in the dictionary for your embeddings?
We already discussed this. So
Yeah, the
we
tokens.
chopped the. Remember, you were the one who explained tokenization.
Yeah, but that was just NLP part.
Okay, the molecular structure is chopped into pieces. We end up with each of those pieces, correct?
Right.
Now, we find the unique ones. We transfer them to the dictionary. Now you have a dictionary of components,
Sure,
right?
but are you showing them? Is it like 2 and 1, or are those?
No.
Oh, the
23,
IDs.
50, 60,
the IDs
yeah.
from earlier.
Yeah,
right?
yeah.
So you map from the IDs
So
to.
word IDs, we get the word IDs. We turn them into embedding.
Calling it a word ID is so counterintuitive.
It's not my fault. The authors have used it, and I think it kind of makes sense.
So just to make sure we got this, so you go from 2 to leaf.
No, that's.
So because that's where he gets the IDs, right?
So
Yeah,
we're
it's
collecting
climate.
information about each of these nodes. This node has a word ID, right? It was one of the components of the structure.
2, for example, has an ID. Yes.
So we get that. It's assigned to a specific structure. We have that.
get
We
the
transform.
embedding. Okay,
Does it
right.
make sense?
Yeah.
So that was just how they make us.
Now, what type of information are we transferring? So basically, initially we have no information. So we start at, let's say we start at a node. Here, this leaf. What information does it contain? Zeros. So what they do is that they collect that information, basically a tensor of zeros. Tensor of zeros, and then they combine that with the word ID, with the embeddings, not word ID, with the embeddings, and then they feed it to the GRU. They feed it to the GRU. GRU now, what does it do? It gives you new memory or the new message.
What does GRU stand for?
So there are a few types of recurrent networks. One of the simplest structures to get very efficient ones is GRU. Basically, they're one of the best in sequential classification, which means predicting the next item in the sequence or classifying the next item in the sequence.
Supposed to be the evolution to LSTM.
Yes, but based on what I understand, with smaller data sets, GRU works better.
Less parameters because
So we send the information to GRU. GRU gives you the new message. We started with no message or no information. Now we get the information for this edge. We move up the structure and we collect the information. Now let's take a look at how this process works. You start at 2. It skips neighbor 1.
We can't see.
Maybe stay on this side.
Okay. So
Sorry, that's fine.
you start with 2. It skips neighbor 1. Why? Because it's collecting word ID for 2. It collects the information from this edge, doesn't have any information, 0, 0, sends it to GRU, moves up, collects information from this edge, gets the word embedding for this, sends it to GRU, collects this, and then continues doing this. So when you're collecting information, you're collecting that information from all the neighbors. So these are considered neighbors of 6. So if you're calculating this, you will be transferring information from these and also the word embedding for 6. Now, you do a bottom-up and top-down scan. You transfer the information, you end up with the eventual message. Now, the final messages that you get,
So do you do one after the other, or like?
they do bottom-up and top-down, and then they stop.
Do they use that output of the bottom-up to feed into the top-down, or do they still start with a tensor of 0?
In one case, I think they use that. So I think when calculating 4-5 or 5-4, they end up using something like, so it doesn't necessarily only use like one way that the arrow goes, like it's not always this way. It sometimes has to use information from the other side too. That's why they first, I think that's why they first do bottom up and then they do top down.
So 2 and 3 both start with 0 tensors.
Wait.
Right?
2 and 3 start with
2 and 3 start with 0. Both of the. Yeah. Right.
This starts with 0. This sends information. This has to start with 0, but GRU gives it a new message.
Right. Yeah.
Okay?
Every leaf basically starts with 0.
Yeah. Every end leaf.
Does every node have a GRU, or like a separate GRU associated with it, or is it a single GRU and you're just routing it?
So I think it's a single GRU, and you
That's what I
keep
thought.
sending messages to it and collecting new
And
messages.
is it like pre-trained? Like or is it parameters you set to it? Like how does this GRU decide what to do to this 0 tensor after you
Initially randomly outguessed and then it learns.
Yes.
Okay.
Okay, so you ended up, we ended up doing one bottom-up scan and one top-down scan, and those are the eventual messages that you have for each of the edges, correct? So what do we do next? The next step is to figure out what I think is the belief for the network. So you calculate the message for the node, for the root. So you add up the messages that are next to it from the neighbors, and it's word embedding. You combine those, ReLU, and you end up with the overall message for the whole tree structure. Good?
Stupid question. So the overall message, what is it? Like, what is, how do I calculate the loss function? Presumably there's like one label after I train on one tree, one molecule, and then it'll give me some loss function. What is the physical significance of that? Is that like how likely this molecule is to bind to the right receptor, or how?
So they end up this is like, we should be discussing this later, but they end up training the model and then using another model, using the train model to train another model to figure out the different properties. So right now, we're technically just learning the structure. We're just doing that.
So we're making an embedding basically. So that's the end goal.
This is like a message passing algorithm, right? So usually what you're trying to do is to learn embeddings for each of the nodes in your graph. Like one typical way to do this is that you start from some random embedding, which could be just IDs, and you start doing these message passings, and you just keep repeating that until you converge to an embedding that doesn't change anymore if you keep doing it. And using GRUs is a way to achieve that convergence.
Cool.
But in the standard technique, it is you're using. Yeah, sorry. In the standard technique, you would be using more than one GRU, or a single GRU again.
I don't remember. That's the part I don't remember. Because a while ago we presented a paper that used a very similar technique to learn representations from source code, which is essentially the same. You just grab the graph from the source code and you do this whole message passing thing to your representation.
I would say that's a design choice.
Yeah.
For a specific algorithm.
All right, so we're basically done with the junction tree encoder, and we're going to move on to the molecular graph encoder. If you thought the previous process was complicated, wait for this one. Okay, so this process has two steps. In the first step, they create the molecular graphs, and the information they create can be divided into features and graphs. That includes atom features, bond features, atom graphs, and bond graphs, and something called scope, which for a batch stores the number of atoms in each molecule. Okay, so we start with atom features. So technically, what they're doing is basically now collecting information about each of the atoms. What type of information are they collecting? The symbol of the atom, number of bonds, formal charge, chirality, whether or not it's part of aromatic ring. Don't worry about what that information means.
So that's all the features they use?
That's all of it.
Okay.
So that's it. Just consider that they're collecting information for each of those atoms. And they map these based on a dictionary of values they have and transform it into a tensor. And they continue doing this for all of the atoms and collect the information, and they refer to it as atom features. Now,
where's the starting point of those? Does it start from smiles?
you already have the information. So when you were performing junction tree, I think we also stored the structure. So they're transferring the information. So that's how they're using it. They're not using the junction. This part doesn't use the junction tree. This just considers the fact that this is our structure. It's going to start collecting information. So the junction.
So that's the process graph, right? Is that the
This is the molecular structure.
Right.
So-
I want to say if it's the pre-processing stage, or it's after you process and did all the grammar.
Oh, after we had transformed smiles to. You remember I showed the pandas table.
Yep.
So consider that pandas table as the information they collected for all this process. They collect a lot of information. Like I probably showed half of the amount of information that they
So that's
collect.
the most compressed version of the graph, right? Is that correct?
This is referred to as a molecular structure, which represents the molecule that we fed into the model. So each of those numbers represents atoms or carbons,
Yeah.
and this is our structure.
And that's the full graph. All I'm asking is.
Yes.
That's the full graph.
Yes.
Okay. Yeah.
I
Those are actually the atoms and the bonds.
The original. Okay.
So
Yes.
yes. The outputs, the final output of the whole junction tree process is being fed into here.
No.
No, we
No,
forgot about
this is
that.
what we're starting from.
It's
new
just completely
part.
new and different?
Yes.
No, we're
All
starting
right.
from ground zero.
Yeah.
Well,
This is
something
the
like
molecular structure.
Yeah.
So what the
What did we
output of
do
the-
with the junction tree?
Yeah.
We're keeping it.
So is it a representation of this? I just want to understand, like, what does that
Wait,
represent?
this process, as I mentioned before, this process is going to be more complicated than before. So don't worry about the
Okay,
junction tree
okay,
for now.
okay. I'm waiting.
Okay.
Horses.
Now they collect information about the bonds. And the information they collect is simply the type of bonds, so single, double, triple, and also their geometric properties. Don't worry about that. I'm not going to explain it. Just consider the fact that they're collecting information about these bonds. So going one way and the other. And they end up with a tensor of the features. Now, the way they collect this information is a bit different than how I would do it. They collect the information for each of the atoms. So you have a bond that is 0, 1, but they collect the information for both of them. So they include the information for 0 and they include the information for 1. And they put everything together and end up with this monstrosity.
And so it's all, you have two of the same all the time? Like,
So their design requires them doing this. They later on end up using the information here, and that's exactly why they collected like
And so
the
is that
information.
because- I'm assuming it's a bidirectional connection?
Yeah.
Is that why?
Yes.
Okay.
Later on, they're going to create that. Let's see, the next step. So you remember how before when we were creating the junction tree, we talked about the fact that information needs to be transferred in the graph. Now here, they create atom graph. And this is a method for transferring information to the atoms. So if you pay attention to the color coding I have here, so the bonds color coded as yellow, they are the ones that are connected to atom 0. So they're collecting information about which bonds will link to a specific atom. So, and to do this, they first create a table of for all the bonds possible in the molecule, and they give them a unique ID. Now, the next step is just figuring out which bond is which. So, for instance, 1, 0 is 2, and they transfer that information here. Next one is 4, 0 is 33, and transfer the information. So that's how the process
What
works.
is that? What is in the related bonds?
Oh,
So they find the connection. They have a representation
So,
for it.
Atom
What is-
0
Yeah.
is connected to 3 bonds. 0, 1, 0, 4, 0, 6. Let's. Did you
Okay,
get
yeah,
it?
I get it.
So then they figure out where that bond is and collect the ID. And they transfer the information.
Question.
I just actually want to say, I think you're doing a really good job.
I hope so.
This is nightmarish. I'm so
This
glad
is-
I did not have
Just
to
because
read
we're asking questions doesn't
100%
mean you
on
don't
this
explain it well.
paper.
It just is very complicated.
All right.
How many columns are in this? Just those 6?
What do you mean, column 6?
It's a tensor.
In the tensor.
This should be a tensor.
17 and 6.
Yes, 6
See the
columns.
bottom? 17, 6.
60, 80 by 6.
Very small.
So I'm guessing that's
Maximum number.
4. That's the batch. That's the so I think that should be 1 vector for each
17 atoms.
for each
In the-
atom.
17 atoms
Each one
and
has
6 one, yeah.
So 6 is max.
Okay. Now the next
Yeah,
thing they
totally.
good.
We're just resolving things.
We're good.
But I don't see anything with 6 bonds.
I think 2 does.
No, so
I
What
think
with
2
6
does,
Yeah, that's why.
because
That's why
2,
is there
3,
that
12.
just column of zeros?
Oh, you're asking why there
Yeah.
are
I
6?
see-
1, 2,
Yeah, yeah,
3,
yeah.
4, 5, 6.
Like
Yeah.
the max always
Uh,
is 4.
So this is how they designed their system. They're limiting the number of neighbors to 6. So they're saying the maximum number of bonds that could exist for an atom is 6.
But
It's
now,
not going to be that high, but they're just making sure they have a number.
Just so I don't know anything about chemistry. There can
Okay,
be.
this is getting out of hand. We had a comment here I want to hear.
I
you were-
think 2 has got that many connections.
2?
2's got 4.
No, it's
Does that
So
have
the
12
maximum
12
number,
connected over? Is
the
12
maximum
connected over?
number of connections or bonds a carbon can have is 4.
Okay.
Right.
Okay.
Okay.
Done.
and that's why 2, 6, 4.
Yeah, I see that now.
All right, so the next step is to figure out a bond graph. And what is a bond graph? So previously, for transferring information in the junction tree, we had edges, right? And we calculated the messages for the edges and the neighbors and so on and so forth. Now for this monstrosity, we have to do the same thing. So for bond 1, 0, we figure out which bonds are connected to it. Those are 4, 0 and 6, 0. Okay? So we check the table, figure out the ID, transfer the information, same thing. So their implementation made it super complicated to understand this process, but basically the only thing you have to do is figure out which bond this is and remove it from here, and you end up with the result. Okay. All right, so we ended up doing all of this. The only step left is the encoder. Now, what do we do with this? Okay, so what do they do now? They turn every atom into a node, and they end up with a graph like this. Now, previously, they did a lot of work to transform the structure into a junction tree, but now they have no choice. They have to deal with the structure. Why? Because they want to collect the information for each of the atoms. Now, how do they transfer messages in a structure like this? So what they end up proposing is that we should use loopy belief propagation. And how does loopy belief propagation work? You basically initialize the whole network at once and let it run. And hope to God that it will converge.
Wait,
in this.
How does it learn?
Well, technically it shouldn't converge,
does that not
but
work for atheists? Just saying.
Well,
Yeah, hope to God.
Going on YouTube.
Hope to.
okay,
Quick
so
clip on
No,
that.
but how does it learn? Is it just randomly based? Or?
So technically, based on what I read, this should not converge. For a long time, scientists thought that it's not going to converge, but a graduate student ended up saying, the hell with it, I'm going to run it and see what happens, and it actually converged.
Wow.
And after that, people started using it. So now it's okay to use it. Whether or not it works, it's up to you.
So, yeah, like,
So converge, what is converging?
So basically, getting an end result from the whole network, but for a structure like this, the issue is that think about how you would transfer the information. There's a lot of dependencies. So let's say if you want to calculate the information for this edge, it's connected to too many loops. So that creates a lot of issues. And there's no other way for you to do this, at least not right now. So they end up using loopy belief propagation.
So what you're learning here is embeddings for each of the atoms, right? And those embeddings could potentially not converge if you have loops.
Okay, embedding, okay, okay.
But apparently it does.
They could diverge too. Like they could just change
indefinitely.
Okay.
drastically all the time. But the convergence would be them settling down.
Settling on some
on
set
value,
embedding.
yeah.
Okay.
One of the, yeah.
Good. All right. So here they're basically figuring out. So in the paper, they say they start or initiate the whole network with 0. But prior to what I think is their loopy belief implementation, they actually calculate messages. So I'm not sure if they actually set it to 0 or they actually calculate a message before they start the loopy belief, but they actually do this. So what they end up doing is combining bond features, concatenating it with atom features, and creating a new tensor. And they consider that as messages, but in the paper it says that they initiate the whole networks with zeros. So this part, what I say, take it with a grain of salt. I'm not sure if how the implementation works.
Each atom, they encode a message into it?
So you remember how we were transferring information based on edges? For each of these edges, so one way or the other, they figure out their bond features, combine it with the atom features that they calculated before. They basically concatenate them, and they feed it to a linear transform and ReLU, and they call it messages. I tried to figure out how exactly the loopy belief process works, but based on what I understand, this is the initial messages that they send to loopy belief, but they say they send zeros. So I have no idea about this part. Now, how does the loopy belief process works? We created bond graphs, and those technically give us the ability to calculate messages. So, for instance, if I'm calculating the message for the bond color-coded as green, I can use this table. So I have bond 33 and 35. The previous message that was created, I can look it up and transfer that information. And we can continue doing this for all the bonds. After that, you sum them up on dimension 1. So you're basically saying all the neighboring edges, I'm going to sum them up, put them together, and feed them to a linear transform, and add them to old messages. Now, this is where I think it's possible to use the messages, but feed the network zeros, because you can start with like old messages, and old messages can technically be zeros. So you can feed the model zeros and then feed it this. I'm not entirely sure how they perform the Loopy belief process. Okay, so now, remember how using junction trees, we calculated the messages eventually for the nodes? We're going to do this for the atoms. So for atom 0, we're going to add all the edges that lead to that atom. So I've color-coded them differently so that you understand those are the edges that we're using. So similar to the process we had before, we look up the values in the messages, same process, sum them up, concatenate, and they end up calling them molecular vectors. And in the junction tree, we used the root to figure out the belief from the network. What do we do here?
So they just put these bonds together that lead to one atom?
So yeah, so they calculate the messages. Like the Loopy belief gives them eventually. So they run the Loopy belief, I think, for 3 loops, and then they stop the process. And then they consider whatever message they get from for the edges as the final message. Okay. So then, adding these up will give them the message for the nodes or the atoms.
Oh.
Now the issue is this: how do we figure out the whole belief for this network? Previously, we used the root. We added the neighbors together, and we ended up with the belief. In this case,
it's not a tree anymore.
They basically add everything together and divide it by the number of atoms that you have. So basically the mean.
What does the belief represent?
So let's say the final understanding of the whole network. So let's say, how do I explain this? It gives you an idea of how the different components of the structure think about each other. So let's say one of them, oh, I have to go back. Maybe I will explain this later for you. I have to go back to the tree structure.
It's a number that represents something.
Okay. It's technically. All right, where were we? Okay, so we ended up creating the final results for the molecular graph, and those were the molecular vectors. What do they next? We ended up with molecular vectors on the side of the molecular graph and tree messages and tree vectors for our junction tree encoder. Then they basically find their latent representation, and they say our eventual latent representation is basically when we add these 2 values together. Okay. So encoder, latent representation, now we have to feed what we ended up with to the decoder. Actually, the way they trained this the model is that they initially do a pre-train for 3 epochs, and they set the value beta, which is multiplied by KL loss to 0, and they say this basically trains the autoencoder. I'm not completely sure why they do this, but maybe somebody here can.
That seems a bit low.
Beta is learnable?
So, no, it should be just a hyperparameter. But if you show me the loss function, I'd be able to explain.
Okay, so
more,
we get there.
But yeah.
Oh, wait.
Isn't that the last one?
That's the loss function.
That's the loss function. Okay. So it's usually they're probably using beta to.
They set it to 0 at.
At the beginning to make it things easier.
Yeah, probably. Let me see.
What's top loss? Sorry.
Topological loss.
So they manually vary beta.
Yeah. So beta is just to weight that KLS, right? That does make sense. So KLS is your distributional distance from what you want it to be and what your representation is right now. So between the two. Yeah. And beta is just to weight that. How important that is.
Sorry, KL loss between what two? Oh, okay, the data and the model. Okay.
Yeah, so the models. So if it's similar to autoencoders in general, variational autoencoders, then it is between your depending what are you like with what stage we are, but so there are two scenarios. Let's go through them and we can figure it out. So it's either for your encodings, so how far away what you so say you're encoding, how far away what you just encoded is from the encoding that you've seen previously. If it's for the output, then it would be how far away the distribution that you outputted. So say you're inputting X, you're generating X prime, right? How far those are from each other.
And beta just a correlation.
Correlation? I mean, it shouldn't be. Like usually when you say beta, it means a parameter.
So yeah, but that kind of
it is a
correlation.
coefficient. Correlation.
Because if you look at the type group, like the structure of the loss function,
Yeah.
so there are 3 terms at the beginning. There are some like 2 times the correlation times
Correlation.
I'm not sure.
Well, they,
Yes, that's KL. It is KL, right?
yeah.
No, I don't think it's a correlation. It's just a hyperparameter. Yeah.
We could talk about it later.
So all these different losses, they're just easily calculated from comparing the result of the autoencoder and decoder? Or?
No, I'm skipping things.
Because I was gonna say, like, where did all these values
Some
come
of
from?
these losses will require me talking for another hour.
Okay.
So.
One quick bit, like you had HG. Like how was HG?
Where?
Like how was that included in the ZG? Before that you have HG, right? Yeah.
Yeah,
How
So HTG is for junction tree, and HG for is from our graphical molecular graph encoder.
Is that the belief or?
That's both of them. So these two are both beliefs. This tree message is basically the messages for each of the edges.
All like tensors containing all of the messages?
A tensor containing all of the messages. That's correct. That's bottom up and top down messages. So we have to deal with the decoder now. We start with the junction tree decoder. Yeah, it's a duplicate. Seems like. Okay, so how do they use the decoder? The first thing they do is again so you basically what they do is they feed the junction tree to the junction tree decoder, and what the decoder does first is that it does a tree traversal and collects all the some of the information from the junction tree, and then it removes all of the neighbors. Why they do that?
the decoder is trying to get to the original. It's back.
The
trying to get back
decoder
to the-
compares the results, like the latent, tries to decode the data into the data you started with.
Okay.
Okay? So you started with some molecules, you encode them, the decoder tries to create the same thing. Now, I'm not entirely sure why they do this. They talk about teacher forcing. This might be the reason why they're doing this, so that they can teach the model where it makes mistakes, but I'm not entirely sure. Now, how does the decoder works? The decoder starts with generating the root, and then the process is similar. It's very similar to the junction tree process, but this time you don't have the whole tree structure. So in each step, it adds a node, it calculates the message, it labels it, it figures out whether or not that's the final node. So this is what they refer to predict stop. So some nodes should technically be leaves, right? So each time it does the calculation, it figures out whether or not that's where the structure has to stop. And they do this for the whole structure. Now, this order that they came up with actually translates to this. And the way it works is that it starts moving down the structure until it reaches a leaf, and then it backtracks. Backtracks and figures out the nodes. So based on what I understand, they basically move from the root to the final leaf, and then they move up and generate other nodes.
The first search.
The colors change again. Just want to make sure it's not on purpose, right?
Which colors change?
Like four and
so
five.
This is the decoder. The decoder is going to forget what it was dealing with.
Okay.
Okay?
So it doesn't matter.
It's going to just use the latent representation to recreate it. So the best way is to tell it to forget about what you're dealing with. This is just the latent representation. Try to get to the final molecule based on the latent representation.
It just has
So
these GRUs that.
similar to, because we used that for the junction tree, we have to use it here too. That was like for the encoder we used it, for the decoder we have to use something similar. Now, the final step is to use the molecular graph decoder, and this process is basically so complicated that I gave up on trying to understand. Okay, how does it work? They basically feed the junction tree, the tree messages, and the latent representation for the molecular graph to the decoder. The decoder then finds all the possible nodes from all the nodes that are possible for that situation. And then it starts enumerating, and it combines them together and finds different ways of combining these substructures. And for each time it creates a new structure, the structure gets scored. And based on the scoring, gets rejected or it gets accepted. So this is the process. How the process works. These are the components that they start with, and in each step from here, so that's a leaf, they combine these two. The combination of these two could be different. They can connect like this, so the sulfur can be here or the sulfur can be there. So you end up with two structures.
And how do they compute or get to those two?
What do you mean?
You're saying they take these two, the decoder, but how do they get those two from where?
So from the latent representation, from the mole tree, and from the tree messages. They feed everything into this molecular graph decoder.
It just spits out an answer?
Yeah, this is the implementation is similar to their encoder, but one thing, I will talk about this later. One slide, I think there's one slide left, hopefully. I'll talk about this in the next slide, but there's a reason why they're feeding everything into this decoder. And the reason is this. and it's super complicated, and I'm not sure if I fully understand it. But here's what how they explain it. So previously, we just used messages from the molecular graph for creating the belief. But in this case, they're not only using the messages from the molecular graph, but they're also using the messages from the junction tree. So this is a bit different. So they're combining the two messages. And they explain how the process is done, but I have no idea how it actually works. With that,
well, if you're just combining messages, probably some linear and nonlinear interpolation of.
So it's actually this is me trying to explain how it works. So you know how in the molecular graph structure we had the actual structure and we use that. I believe what they do is that they use the tree messages and figure out which bonds connect the structure that were represented in the junction tree. And then they insert the messages from the junction tree into the molecular graph. That's what I think. I'm not 100% sure. That's the best way. Like that's the best idea that I had to solve how they do this. Now I'm going to conclude with this, but they have done a lot more work, but it's out of the scope of this presentation. They have actually done a lot of experiments. They got very good results when it comes to the validity of the results. So they actually got 100% validity of their structures. And when it comes to reconstructions, the reconstruction of the structures, they got 76.7. And when you compare the validity to other methods or models that were developed before, you get a very good result. And the main reason behind that is using junction tree and also the method of combining substructures together. So this method of combining substructures together has been very beneficial in this model. So with that,
these substructures are, can you just, what are you referring to when you say these substructures?
remember we chopped the molecule into little pieces.
All right. Oh yeah. Okay.
Okay.
Don't go back.
Substructures.
Everything they did before Autoencoder.
Got it. Yeah.
So substructures.
Yeah.
So they did this.
It's like a lifetime ago.
Yeah. This
Now
work
you're just
is-
making fun of us.
This work is actually really good, and I don't think I have done it justice. I've tried my best, but I enjoyed reading the work. With that, I thank you all for.
Since we had a lot of discussion, let's ask 3 questions, and then we're done.
No pressure.
First, thank you for the awesome presentation. In the second part, you didn't mention that what type of the decoder we are talking about. The first part is the group, right? I said the GRU and the GRU. But in the second part, what type of decoder do we have it here?
What do you mean?
Yeah, here. Here, what's the decoder actually?
They also use GRU. In this case, they also used. I'm just skipping stuff. They use GRU, and this also I think is based on Loopy belief.
Sure. In the part that you talked about pose and then you want to break the loop, so you end up with number 6, and I believe that in your slide there's a loop in between 4 and 5. Why you want to break that loop?
You mean in the molecular structure there's a loop?
Yeah.
So this is actually based on the 1998 paper by Dixon, and they make the assumption that cyclic structures that are connected with minimum of 2 carbons can be connected together and assigned into or transformed into one node.
The tree that they used at the very end didn't have any loops. They removed
Yeah, but before that, when he he breaks the loop, and then he breaks the loop and make the node number 6. And but there was a loop between the node number 4 and 5, but he
Maybe it's 'cause they had less
left
than-
that one there.
Do they have less
He
than
removed
two?
the node, he removed the link from that one too.
Yeah, later that one's happening.
Yeah, we do MST.
That's because I think it also it had less than 2 common atoms. Right? Is that right?
I hate that.
He's just tired.
I'm done.
Okay, last question. Somebody who hasn't asked any questions.
Yeah.
Okay, you don't have any? All right.
I can ask a question quickly.
Yeah,
So your last slide of conclusions was it's 100% valid. It's really easy to check to see if your molecule is valid or not, right? Why can't you just generate a lot more invalid or like just generate with some of the other methods and just throw out the invalid ones?
Like, you're saying generate lots of invalid ones and select the ones that are valid?
Well, then you
That's
need,
just
like,
extra work. Why would
Probably
you
because
train a model that would generate a lot of invalid
crap
results?
and then just filter out the crap.
I'm just guessing wildly, probably because there would be
No,
too
no.
much
So
degree of freedom.
let me explain it. Let me explain it differently. So these are actually the results. Compared to the results that the previous scientists got, these are actually very sophisticated structures. So compared the results that the previous researchers got have molecules that are invalid and have molecules that are in no way as complex as these ones.
Just to add to that point, I work with generative GANs a lot, and there's a similar sort of issue. So you, basically, there's always a problem of how are you actually going to evaluate one of these models, right? You're generating, in theory, new molecules. From what I understood, they're actually checking them relative to an actual data set of possible models. So what they're looking for is how well do the models actually line up with that data set. And what they're saying is every single molecule that was generated did have a corresponding real molecule in the data set that it matched up with.
Yeah, I think his question was about like what's the cost of generating too many
invalid ones?
Yeah.
Well,
And also, I think
okay,
this is
so-
important too,
so-
like there needs to be ways to measure the success of any approach
Right.
like this, and
So-
you need something like that, like to compare to a real chemist would come up with this. They say this is great.
Well, because again, the end goal would be that you don't actually have a database and you're just generating molecules on the fly that have never been seen before. So invalid means that your model in some way doesn't properly capture the underlying embedding space. I think that's what they're trying to do.
That is correct.
But after going through, how many layers of feature extraction was this? Like, if it was less than 100%, I would be, yeah.
Pretty mad.
I would